So for somebody who spent the past 2 decades of his life working on clinically directed gene editing, the only thing more overwhelming than speaking after Eric Olson is speaking after Eric Olson and before Peter Marks. With that, I'm here to speak with you about the notion of going with a CRISPR medicine from an N of 1 to an N of all. Neil described sickle cell disease as a genetic condition in 1949, and 70 years later, the first human being was apparently cured of her sickle, and her name is Victoria Gray. She's a brave human being. And Dana showed data about her in his opening talk. She has had no vaso-occlusive episodes in the 4 months post-gene editing with CRISPR. And the data that describe the biological basis of her new state are truly, truly remarkable. Approximately half of the hemoglobin in her bloodstream is normal. I want to show you 2 slides that highlight how this was done. Because, in fact, Victoria Gray has not had the sickle causative mutation corrected in situ. And therein lies an important lesson for what I'm going to tell you today about the age of the druggable genome. I want to show you 2 slides that you saw. This is from Jennifer Doudna's talk last night. And she highlighted the fact that you can interrogate the human genome at many scales. You can poll the entire thing in a genome-wide association study, or you can ask about the function of individual base pairs, such as the 1 mutations in which cause sickle. I could not have gotten a better introduction to this section of my talk than the presentation by Zach Lipman, which is on called Persicon esculentum, which I renamed Licas persicum, which is basically what Zach has made this organism into. And what Zach has shown quite beautifully is, thanks to genetics and the use of Cas-CRISPR, you can create a continuum of engineered trait variation. And the way that is done, the way you create a rheostat from small tomato to large tomato, is you gene edit regulatory elements that control the function of genes that are involved in this trait. Why am I showing you this? Because this is precisely what was done to Victoria Gray. It will be immodest of me to spend any fraction of my talk describing work that my team did at Sangamo Therapeutics to make this happen, so I won't. Except to say that as a result of that, and the discovery of this target, a particular transcription factor binding site in a regulatory element that tuned up a protective gene that is really what's running Victoria Gray's erythrocytes right now, the fundamental framework of discovering such targets has now been established and involves starting with the genome and looking at a genome-wide association study, then using epigenomics to map regulatory elements that confer the desired level of control, and using a sophisticated combination of low-resolution and high-resolution genome editing to finally get to a target that you can engage in the clinic, as was done with Victoria Gray, and CRISPR Therapeutics and Sangamo Therapeutics are both pursuing in the clinic right now. What have we learned? Well, first of all, we appear to have 2 humans cured 4 and 9 months post-transplant, so early days, of their sickle cell disease and beta thalassemia, respectively. So this is a formidable moment, 7 years after Jennifer's landmark paper. Here we are with an apparent cure. And of course, this was not targeting a kinase or a GPCR. This was targeting the regulatory element of a transcription factor. So say hello to the age of the druggable genome, which is what, of course, the age of CRISPR has given us. This is a meeting to ask real-world oriented questions, so the 3 are. What will it take medically and commercially to scale this to the 100,000 Americans who have sickle? And for genetic disease, the rest of it, what are the implications for those folks who currently have one of 5,000 genetic diseases? And for common disease, we know that loss of PCSK9 is protective against cardiovascular disease, or of SCN9A against pain, or that ApoE4 causes Alzheimer's. But what implications does any of this have for the future of folks with these conditions? Well, we know that a pessimist is a well-informed optimist. The answer to question number 1 is not good, because this involves a myeloablative transplant, and Peter Marks is a trained and well-experienced hem-onc physician, he will tell you that an ablative transplant is not something you want to do casually. And it's going to cost $2 million if this cure is approved. The demographics of the affected population all add together to the fact that this will be operationally impossible under the current state of affairs. What implications for folks with current genetic disease? Even for monogenic disease that's tractable using today's technology, an average carrier of a point mutation has essentially no chance of being gene edited in the clinic in the next decade. An average carrier, I emphasize, and it's not a delivery problem. And as far as common disease, well, at current trends, an approved medicine is 10 years away. And the rest of my talk will be spent discussing what can be done about all of those issues, and I'm strongly convinced that they can be overcome. One thing I really want to emphasize, and this very much touches on a point that David Zilberman made in speaking with the gene drivers about the fact that inaction costs lives. I think for our field, slow action is not a safer choice. I will show you two pieces of data that prove that. Piece of data number one, there are folks out there who use CRISPR and they don't know what they're doing. I don't have time to indict these folks for what they did, but this clinical trial of one should never, ever, ever have taken place. The reason it happened is it was easy for them to make a guide RNA, but everything else they did wrong. But this was in a hospital, at least this was within some conceptual framework of investigational medicine. I unfortunately, thanks to a request by Antonio Regalado, had to read Zhang Kuihe's set of pieces of paper. I don't want to even call it a manuscript. And the last quote that ends his story about knocking out allegedly CCR5 is, "We here bring a novel therapy to enable acquired immunity and to control the HIV epidemic. We anticipate that human embryo editing will bring new hope to millions of families seeking healthy babies free from inherited or acquired life-threatening disease." The profundity of the delusion and the hubris is overwhelming. But the problem is that Cas9 is too easy to program for use by people who have just as severe a case of delusion plus hubris as Zhang Kuihe. So, there is a book, this is real, I'm not making this up. If you Google CRISPR, if you put CRISPR into Amazon, you get a book called CRISPR Apocalypse, which features brilliant scientists, maniacal Russians, etc. So I told Jennifer Doudna that between her and I, we have the first two characters covered. So we don't need nonsense books on Amazon. The world will not trend the right way unless we act. So I will focus on sickle because this has been a professional and personal passion of mine for a large fraction of the past 20 years. That's, of course, Victoria Gray being treated by the nurses. What are the key inflection points? I think one, and critically, we have to go from viral gene therapy to non-viral genome engineering. We have to get away from an ablative transplant where cell processing occurs ex vivo and it happens in a centralized facility that requires GMP, to a non-ablative, closed-loop ex vivo point-of-care processing, which ultimately transitions to point-of-care in vivo delivery to HSCs. I greatly admire my colleagues at Sangamo, folks at CRISPR and Editas Medicine, and other companies who are in for-profit entities. My strong assessment is for this to make a major impact on lives of Americans with sickle, this will have to unfold in the public sector, and I will argue to you why. In looking at all of this, I get the fact that many of you will say, yes, but X, Y, and Z is not working yet. I'm here standing with 14 years of experience genome editing, actually 15, to tell you that eventually the technologies will get there, and I'll show you two pieces of data from my personal experience. In our first description of a correction of a native gene, there are many pieces of data that, frankly, I'm proud of, and Mike Holmes and Phil Gregory and Ed Rebar and my colleagues at Sangamo should be, except this, we tried to gene correct a primary human T cell, and we got this miserable band, which I had to quantitate. The cells basically died. Exactly 10 years later, Jinping Wang and Michael Holmes at Sangamo solved all of those problems and got to 56% repair of a mutation in T cells. Thank you very much. And what does that mean clinically? Well, that means that this has completely changed the landscape of how we think about treating cancer. I wish I had a movie of a patient or an animal, and every time I see the film that Eric shows, or Leonella shows, I want to cry with joy because it's like such a moment for all of us. But let me assure you that the bar graphs here are no less overwhelmingly exciting. because what they show is the ability, for the first time in the history of humanity, to perform multi-locus, exquisitely potent and specific genetical engineering of clinically relevant primary human cells at clinical scale production for cancer. The data on one end are Sangamo. The data here are data from, I think, CRISPR Therapeutics, and other companies who work in this space have data like this as well. So that the underlying zinc finger Cas9 tau meganuclease is not relevant. What's relevant is that we can multi-locus edit with extraordinary precision to weaponize cells against cancer. And let me say two things as far as technology pace. If you showed me these data in 2014, I would have called for a psychiatric consult. There is no way this could have worked. And yet here we are. But critically, this was not a magic bullet. There were many iterative incremental improvements that added up to this extraordinary level of potency. Now what? And now begins the real deep challenge. And if you speak with any person who has ever filed an IND with the FDA or filed with the EMA, or anyone who has looked at early stage clinical data, they will say to you that the only way to find out how to edit people better is to edit more people. There are fundamental questions about safety, efficacy, biodistribution, and dosing that you cannot establish in any preclinical model of efficacy and safety, and thus the only route forward is to advance more CRISPR therapies to first-in-human trials safely, ethically, and effectively, of course. But that's the only way forward. That's what Bluebird did, and they have the 1.8 million cure for thal, soon to be probably approved for sickle as well. The way this was done, I don't have time to do justice to how much work went into this. I asked for the slide from my friend Philip Gregory, who is the chief scientific officer of Bluebird Bio. What did they do? They developed a V1.0 product and they dosed a bunch of subjects. They looked at the data, it wasn't good enough. They tweaked a couple of parameters, they dosed a bunch of subjects. They looked at the data and it wasn't good enough, and they dosed a bunch of other parameters, and finally it worked. This is the only way this can move forward. So, but critically, I don't think this can be left to industry. In the discussion section, I'm happy to speak about why. And we cannot wait for the perfect everything before going to first in human. We just can't, because the perfect everything will never exist. So instead, we must build a non-profit model of advancing innovation to first in human, and we must build a fundamentally new way of doing vertically integrated translation in academia. By that I mean end-to-end. So the Innovative Genomics Institute doesn't just talk the talk, we very much walk the walk. We are driven by Jennifer's and Emmanuel's discovery of what Cas9 can do and how to program it, and we are on a mission to use genome engineering to improve public health. I won't speak about the infrastructure, I don't have the time, but I will say two important things. We are at the University of California, Berkeley, and San Francisco. Our motto is Fiat Lux, let there be light, so we're driven by a mandate from the taxpayers of the state of California to have a halo across the state and the nation. For that reason, we focus on what we call end-to-end solutions. And I'm going to give you a few specific examples, not to promote the IGI, but simply describe to you what I think is an essential component of how CRISPR can become an N of all. We work on all pieces of the puzzle: editability, targets, specificity, being able to make specific edits in specific settings. We work on delivery in a major way, and we embed first-in-human trials within the IGI ecosystem. I could give you a 10-minute presentation, if not more, about every single one of these names. I don't have the luxury of time. The way we think about this is the Avengers model. So you have a group of superheroes with non-overlapping superpowers collaborating on the same challenge. Couple of quick examples. Alex Marson and Jennifer Doudna made the landmark discovery that you can efficiently and safely deliver Cas9 and the editing machinery in the form of a ribonuclear protein particle. Then work by Dana, who is here, and really pushed by Mark DeWitt and Wendy Megis and David Martin, with Mark Walters at Children's Oakland, and all of this was led by Jacob Korn. Produced a process where now with funding from the NIH and the CERM, so I emphasize public nonprofit money, we are on track to file an investigational new drug application with the CBER on a gene correction approach for sickle cell disease by the end of next year. Ask me again in 18 months how it went. Why are we working on sickle? Well, in part because we think that the current industry pricing scheme is simply not scalable. So led by Jennifer, the IGI is thinking deeply about how to create a solution where, inspired by the vaccine precedent, to develop, deploy, and protect certain editing-based medicines in the nonprofit public sector. I emphasize the word certain. It would be disingenuous of me, after having worked for 16 years in a publicly held biotech, to say anything untoward about that. But I'm simply arguing that certain indications and certain approaches, there is a national need and a global need to protect them in the public sector. In that regard, I did a sort of a private version of the Macarena when I saw this, that Gates and the NIH have partnered to provide no less than $200 million for a scalable gene-based approach to treat HIV and cure HIV and sickle. And I really salute both organizations and recognize Dan Wattendorf, who's here and who played a key role in this. I am convinced, that success will have to come from a joint effort by the public sector, such as IGI, from the federal government, the CBER, and from philanthropy, such as the Bill and Melinda Gates. There is right now no automated process whereby something that comes out of this initiative or the Somatic Cell Genome Engineering Consortium, which the NIH runs, somehow cures a substantial proportion of Americans with sickle, to say nothing about the 150,000 children in Nigeria. In order for that to happen, current effort has to be forward integrated towards that at every level. And forward integrated means you think now about every problem that arises on the path forward and you start working towards solving those problems now. We walk the walk. And allow me to spend a couple of minutes telling you about an effort that Jonathan Weissman leads at the Innovative Genomics Institute. We have a very generous grant from DARPA to develop a first-in-class radiation countermeasure to protect the American warfighters, specifically, in particular, special forces soldiers whose operators have to be dropped into all sorts of theaters of war, and also folks who are about to go radiation therapy for their abdominal or pelvic cancers, and thus are at severe risk of dying of lethal diarrhea or bone marrow failure. DARPA has funded us in an end-to-end vertically integrated multi-locus institution program, the entirety of which is within academia and the nonprofit sector. Jonathan is leading the way to use extraordinary advances in screening that his group has developed to find targets that, when controlled in intestinal and hematopoietic stem cells, will protect those from radiation damage. Your humble servant, together with Jonathan and with Lou Gilbert and Dirk Hockemeyer, is leading an effort to engineer a CRISPR-based transcription factor, more on that in a second, that would inscribe epigenetic memory of the radiation protective state on those genes. We're very fortunate to collaborate with Katie Whitehead and to have Dirk Hockemeyer's expertise on building for this project innovative, first-in-class, in vivo delivery solutions, both to hematopoietic and intestinal stem cells. And finally, and I really salute DARPA and the Department of Defense for having the vision for funding us all the way to IND. We have received, if we get through all the metrics, we are funded to do the CMC at clinical scale production. We're funded to do all the small and large animal IND-enabling farm tox PKPD, all the regulatory costs, and go to IND and do the clinical trial at UCSF. This is the way to do this. And I cannot, Rene Veggen, who is the program manager at DARPA, I really salute her and DARPA and the Department of Defense for this support. In the few minutes that I have left, what about, so I spoke with you about sort of what it will take to scale up for sickle. What about the N of 1? Well, I think Eric did a beautiful job introducing this. What a landmark moment. I think the identification of CFTR as the gene mutated in cystic was exactly in 1989, so 30 years ago. Vertex, I salute them for developing small molecules that appear to be able to treat and cure 90% of the population. But here is the challenge, folks, and I cannot emphasize this strongly enough. If there's one thing you take away from my presentation, other than the last slide, it's what I'm about to say. This man has cystic. He does not have one of the 3 mutations that the Vertex drugs treat, and he will not live beyond 40. Why can't we just CRISPR him? And folks will say, oh, well, it's the delivery to the lung, to the ionocyte. It is not just that. You cannot see this on purpose. These are some of the 1,500 mutations that cause cystic. And even if you could deliver to every ionocyte in the lung, repair of every single one would require a different guide RNA and a different repair template, and that is in UIND. And that's in U farm tox, and in UGMP CMC. And we have a magnificent opportunity, given the outstanding history of collaboration with manufacturers, with CMOs and CROs that provide animal services, and critically with the Center for Biologics Evaluation Research, to make an impact. I am thrilled and really salute Timothy Yu at Children's Boston for the absolutely heroic effort that they have undertaken in building an antisense therapy for one child. As they themselves acknowledge, it remains it's not suited to the treatment of other patients with Batten's because its design is customized to our patient-specific mutation. I was going to talk a lot more about this, except of course our next speaker is the head of the CBER and wrote a magnificent editorial about this landmark moment. And I'm just not going to say anything because you're going to hear it from Dr. Marks himself. What is the IGI doing? We are particularly interested in disorders of the immune system, thanks to leadership by Alex Marson. Again, talk about basic science innovation driving first in human. Alex's group developed an extraordinary way to repair mutations in primary human T cells, and we have that human. I don't know her name. I know she's 15. I know she's Kevin Harold's patient at Yale, and she has a mutation that causes severe autoimmunity. And the IGI has philanthropically fundraised to do an N-of-1 clinical trial for her by repairing her T cells. Ask me how it goes in about 18 months. But this is not scalable. We cannot keep raising, millions and millions to save individual children, much as we want to. I made this slide for a keynote at a meeting of non-editors just to highlight how truly simple the rules of CRISPR-Cas targeting are. But that simplicity is deceptive in the sense that that's only part of the puzzle. And the IGI wants to provide solutions for all of the puzzle. Specifically, to go from patients to variants to effector to delivery to efficacy to clinical trial and back to more patients, you have to engineer the effector, and that's where CRISPR-Cas comes in. But there's so much more that needs to happen. And we're building all of that at the IGI, within the IGI ecosystem. So a platform to do reverse and forward genetics in primary cells to understand the causative variant and the biomarker of efficacy. To be able to build effectors to not be limited by the design density of Cas9. We're passionate about delivery, and we are both philanthropically and federal government supported to build new delivery tools. We're building turnkey modalities for engineering efficacy and specificity. We carefully choose paradigm establishing programs, and of course, we're ramping up to do the clinical trials at UCSF. The IGI is the engine of this, but I cannot state strongly enough to what extent I am excited to partner with the CBER for the next decade of this kind of effort, because for the past 10 years, as somebody who was there for the very first gene editing IND in 2009, the only reason we got as far as we did is because the CBER was an essential and constructive partner in making sure that we do this safely, ethically, and effectively. Last two slides. I think common disease is going to be a really interesting challenge. I think it's tractable. We have good targets, PCSK9 for cardiovascular, we have tau for dementia, we have NAV 1.7. So I think the age of CRISPR has really made everything druggable, as I have argued to you. My personal bias is for most forms of in vivo engagement with the genome whenever possible. I think rather than do genome editing, I think we should consider doing epigenome editing. And this we owe to Jonathan Weissman, who is right there, and he invented CRISPR-I, CRISPR-A, which is a technology that allows you to tune genes up and down with exquisite precision and sort of rheostat-like tunability. This is just in terms of farm talks, I would be more comfortable deploying an epigenome editor than a genome editor in vivo, at least for now, certainly for common disease. But the fact that we have new technology, and this is my last data point, doesn't change the fact that technology is just the first part of the puzzle and that we will have to rotate around this circle. Somebody will have to drive that rotation, and we have to have we will collaborate at every step with the CBER because nothing happens without proper regulatory oversight. So last slide. Right now, the 1.0 edited human is severe disease. That's what's in the clinic. We're about to start. Cancer, hemoglobinopathies, blindness, hemophilia, liver disease, lysosomal storage disease. 2.0, I think, is we take rare disease and we create a framework for treating rare diseases scalably, and we develop delivery that goes beyond blood and liver. We get more subjects dosed, and critically, the sponsors will be able to provide the FDA, the farm tox and the CMC reviewers, the clinical reviewers, PK, PD, and efficacy data from an action of a range of effectors across a range of tissues, across a range of targets from first-in-human trials, so that the regulators and the clinicians and the scientists learn more about how to scale this up. And only then can we go after common disease, which is why this is empty. I went over time, so I'll run through this for the last slide. Leonardo proposed the flying machine, which is all the way here. And then, of course, now we have commercial air travel. A lot of technological advances and a lot of effort of a lot of different kinds of people went into making this happen. I mean, we're at the generation where this can happen to CRISPR. And it's going to be hard, and there's going to be failure. And I pray that, no, I don't pray, but I keep my non-zinc fingers crossed that, there's not an SAE, because one SAE and we know what's going to happen. So I think we just have to basically be brave about this. And what is courage? I have a military background, so I really love this quote from the United States Marine Corps, which I think should really inspire all of us as we advance this into the next decade, that courage is really not going into a room full of fear and afraid. Courage is being afraid and still going in. And that very much, I think, should inspire us to take it to the age of the editable human. Thank you.